Welcome to Brain Ponderings. I'm the host, Mark Matson. Today it's my pleasure to welcome Professor Jason Shepard. He's actually, I think, still associate professor. Uh,
Just became professor a few months ago.
Congratulations. You
Yeah,
need to change it on your website.
that's true. I do.
Doesn't matter, I guess. Um, in the neurobiology department at the University of Utah School of Medicine, Salt Lake City. He's done some fascinating work on a protein that I knew about a long time ago. Pretty much everybody did that studied synaptic plasticity and learning and memory, but he's developed this intriguing story of its viral origins, and he's exploring not only its roles in plasticity, but in neurodegenerative disorders. So that's what I'll talk with Jason about today. Welcome, Jason.
Yeah, thanks, Mark. It's as we were saying, it's been a little while since we've chatted before.
Yeah. Yeah, I think it's like 2005 or something like that, or
Yeah,
six.
exactly. I was a graduate student at Hopkins, 2003 to 2007.
Yeah. And so you grew up in New Zealand. I remember that. Um,
Yeah, so I was born in South Africa actually, and then my family immigrated to New Zealand when I was 14. So I went to high school and undergrad in New Zealand, so at University of Otago. And then spent a year as an exchange student. I did my honors thesis abroad at UC Irvine, initially with Oz Stewart,
I
and
remember
then
that because we helped a little bit by sending some mice to Frank LaFurla, and then
yeah,
you helped with. You want to briefly mention that mouse model of Alzheimer's because it's widely used.
yeah, exactly. So I worked with Frank's lab to characterize that triple transgenic that he had developed, where it was the first model that had both amyloid and tau pathology. But I was mostly interested in synaptic function and synaptic biology. And so I said to him, like, I'm happy to try and do some electrophysiology on these mice. Um, and I was like this weird sort of exchange student, undergrad slash doing graduate work. So they didn't really know what to do with me. So they were like, sure, try, give it a go. And so I commandeered a rig from another lab, Roger Mithorati's lab, and then found someone in Gary Lynch's lab to teach me how to make hippocampal
Oh.
slices. Um, and did LTP, long-term potentiation studies on those mice. So yeah, so all the electrophysiology in that initial characterization paper was mine.
Yeah, and that paper has been cited a lot
It
because a lot of
has.
people are using it, so they have to cite
Yeah, exactly.
it.
Yep.
Okay, but then you came from the West Coast to Baltimore.
Right. Yep. So I went to Hopkins to work, in molecular neuroscience and joined Paul Worley's group and Rick Huganier. So I was sort of a joint student. And even at that point, so like Irvine was the reason for going to Irvine was that I was interested in ARC. I'd
Huh.
basically written I was doing an advanced undergrad course where you had to write an essay. And I had chosen protein synthesis, local translation,
Mhm.
because that was kind of hot at the time. Um, and Aaron Schumann and Paul had sort of published a bunch of studies that looked like local translation was something real. Um,
Yeah, so that's the idea. So neurons are complex. They have dendrites that branch a lot and an axon. And for a long time, it wasn't really recognized that there are messenger RNAs encoding proteins way out in the dendrites, nor importantly that that message can be translated into protein. So you can get, but it makes a lot of sense, right? Because it seems to be waste a lot of energy to have to send some signal all the way back, then you get the messenger RNA, then you got to ship it out, and the
exactly.
synaptic plasticity changes were occurring on like a time scale that's too short to, you know, explain how these proteins are increasing. out in
Yeah,
the dendrite.
exactly. So there was this idea. I mean, so Oz Stewart, who had moved to Irvine a few years before I got there, had pioneered this observation that you can get ribosomes out in the dendrites, and so that sort of suggested that there should be translation capability out there. And then when Paul was studying immediate early genes, so these genes that are turned on with neuronal activity, they had made that striking observation that our mRNA can get trafficked out into the dendrites, and so that was one of the first examples of that. Um, and so those studies had sort of just come out when I was looking around for essays and interests and stuff. So that was cool. It didn't quite work out with Oz's lab at the time because he had just moved and he had sort of changed focus to looking at spinal cord regeneration. So that's how I sort of ended up working with Frank. Um, but I was always interested in learning about these. Ultimately, it was sort of trying to figure out how memory works and how cells store information. And so that was why I chose Hopkins, and it sort of was just a good time. So Paul had done a bunch of work trying to really characterize these mid early genes, and they had built a bit of repertoire of biochemistry and molecular tools, but they didn't really have any functional assays, or they didn't really know what the protein was doing.
Yeah.
Um, and so I went to Rick's lab to learn how to do some of these AMP receptor trafficking, these glutamate receptors, and then it turned out that one of the functions of ARC was to regulate those AMP receptors, and so it's worked out pretty well.
Yeah. And yeah, so why don't you explain that? Because I think when we get to retro, I don't know if you know, so I had, when we get to the viral origins, I had Josh Dubnow on recently to
Oh, cool.
talk about retroviruses and and the flies.
Oh,
So,
yeah. Yep.
so that makes
Right.
it, that was just maybe a month ago or something.
Okay.
But anyway, so right, so go ahead with the ARC story then.
Yeah. So at the time, we knew that there was this sort of induction by neuronal activity, trafficking of the RNA, and there was sort of some pioneering work by John Gazowski, um, with Jim McGaugh and Carol Barnes, looking at, if you blocked synthesis of ARC, you can actually had deficits in long-term memory and long-term plasticity. But that was pretty much all was known. So I was sort of very interested in the cell biology, biochemistry, and functional sort of regulation. And we had a clue. A postdoc in Paul's lab had done a yeast to hybrid screen and pulled out these proteins that are involved in endocytosis of receptors, endophyllin and dynamin. And so the obvious hypothesis was perhaps ARC was regulating these AMPA-type glutamate receptors, which we knew were important for trafficking, important for the expression mechanisms of synaptic plasticity. But the biggest surprise at that point was that we knew strong stimulation, LTP, long-term potential-like stimulation, would induce ARC expression. So we expected that its mechanism would somehow relate to that. But instead, what we found was that when you have high levels of ARC protein at the synapse, you actually get a decrease in the receptors. And so it looked like it was involved in long-term depression. And so we and other labs sort of showed that. Actually, Roberto Malino and Holly Klein's group were working on this as well. And so we ended up having, like, I think it was 3 or 4 ARC papers in Neuron and in the same issue of Neuron.
That's good. Yeah.
Yeah, it was, I mean, as a student, it was sort of a fun exercise, like getting all
Well, it's nice to have multiple labs kind of confirming, similar results,
right.
it certainly strengthens the likelihood that it's right.
Yeah, exactly. I tell my students that all the time. It's like it's actually good if other labs replicate what you've done.
Yeah. Yep. Okay. And right, but still, so you have our when synapses are active, its levels, protein level goes up in dendrites. It's involved in removing the AMPA receptor, so
Yeah.
decreasing the responsiveness of that synapse to subsequent stimulation. But, the immediate early gene products, other ones, I mean, so what led, I guess, just the sequence of the gene led to the discovery that it's may somehow be a ancient viral remnant that kind of got into the genome.
No, so yeah, I could tell that story. So, after PhD, I went to Mark Baer's lab to look at ARC's role or look at plasticity in vivo using the visual cortex. Um, and after that, I moved from MIT to University of Utah to start my lab. And, there were some obvious low-hanging questions there about more detailed explanation for how ARC regulates plasticity, and then ultimately trying to connect it to memory. And but at the time, I wanted to do some real biochemistry, and there was a lab here, Adam Frost, who was an expert on these endocytic proteins, dynamin.
Mm.
And so we wanted to do some in vitro experiments and purify the proteins. Um, and so we started doing that. In fact, my first technician, Nate Yoder, took this on. And the first thing was just to, purify ARC protein from bacteria. We were using standard GST affinity purification protocols. And that's when we started to get these really odd results because we got nice expression of the protein, but then when we wanted to clean it up and purify it, we ran it through a size exclusion column that separates proteins by size. And none of the protein at that point was coming out as a monomer or where we expected it to come out of the column. It was crashing out of the column and in the void fraction, which was odd because there hadn't been any inkling that this protein could aggregate or oligomerize. Um, and so so we just thought we were bad at making the protein. And Nate tried a whole bunch of different buffer conditions. Um, and then finally, he was like, "Well, um, I want to learn how to do electron microscopy from Adam. Why don't I just take a look at the protein because it'd be good sort of practice." And again, we weren't expecting to see anything. But literally the first images he got to scope, there were like these soccer ball structures on the grid, and we're like, "What the hell are these things?" And so that was the observation that led us down this rabbit hole, and it turned out that these large structures looked like viral capsids.
Right,
Um,
so you didn't dismiss it as schmutz
yeah, we could, and
on the
that's where I
grid.
think we were persistent, where I was sort of like, "Hey, this is a cool observation. Maybe it's an artifact, but let's at least see if it is."
Yeah.
And it did take us a while. I mean, at the time, I of course wasn't thinking about viruses, and we actually thought initially maybe it was these clathrin-like cages that were involved, like maybe it was a new kind of coat.
Ah, yeah.
Um, but eventually, Nate and I, so we just happened to have some good colleagues here in biochemistry that were virologists, and one of them is like a National Academy expert on capsids, viral capsids. And so we showed him the pictures, and he was like, "Oh, I think your bacteria has a contaminant. It looks like a virus." And no, we're like, we did mass spec. We know for sure that these objects are just ARC protein. And so that's sort of what led us down. And then when we started to do these bioinformatic secondary structure predictors, they also pulled out these capsid domains from viruses. So it was all starting to make sense. And in fact, Paul Worley had actually collaborated with a group at Hopkins to try and do a crystal structure of parts of ARC.
Hmm.
And they found indeed that there was a fold, a secondary fold that looked exactly like the capsid domain from these retroviruses. So it all started to come together. Um, but yeah, I mean, of course, then the puzzle was why does this brain or how does this brain gene have a retroviral uh, domain or biology? And so we
And
collaborate.
the first question to come to my mind is, why isn't this arc just accumulating in neurons and dendrites and damaging them or something like that? You know,
Yeah,
thinking like amyloid or synuclein or something like
yeah. Yeah, I mean, so that subsequently, we think that the control of when and where it forms the capsid in the cell is quite regulated.
that.
Um, and it could be post-translational modifications that also regulate in other binding proteins. So we've identified a few binding proteins that we think can facilitate assembly of the capsid and release of it. But you're right, you don't really want to have this
Yeah.
expressed for long.
And this backing up a lot. So ARC is in neurons. Is ARC in protein found in cells throughout the body and different organs, or is it mainly in neurons?
It's mainly in neurons, and even within the CNS, it's not really that highly expressed or not expressed at all in glia. There are a few populations of cells outside the brain. There's some sort of dendritic immune cell, um, and perhaps a few other populations of cells, but mostly it's in the brain.
Yeah. Okay. All right. And so let's go to the. All right. So, but by that time there were already ARC knockout mice, right?
Right,
Mice in which the gene had been knocked out. What's the phenotype of those? You mentioned there's evidence involved in memory,
right.
but then it has, as far as not apparently promoting LTP, strengthening of synapses, it's kind of promoting the opposite. So it seems kind
Yeah,
of like a counter-regulatory system
right.
in plasticity.
Right, exactly. So we had shown in the ARC knockouts that both long-term depression and synaptic scaling, this homeostatic plasticity was disrupted in the knockout neurons. And in the mice have you know behavior-wise they're pretty normal. They grow up normally. They look normal. Learning is intact. So for most kinds of learning that we've run them through, if you look at immediately after training, they're fine. They can recall the information. But if you come back a day or more later, that's when you start to see these deficits. There's in recall. So we think it's in that consolidation process, making short-term into long-term memory, that's disrupted, and sort of long-term forms of experience-dependent plasticity as well in the visual cortex.
All right. So it's but is the formation of these capsid structures in any way involved in its effects on synaptic plasticity?
Yeah, so that's the big question, right? So, like, of course, we'd always expected or thought that ARC was involved in this sort of cell autonomous plasticity. But this idea that it forms this capsid suggested that perhaps it retains some of these other properties of retroviruses, and that includes, um, being released from cells and then being taken up by other cells. And so we showed that ARC indeed does get released in these vesicles. They sort of invaginate out from the membrane, and they can then get taken up by neighboring cells. So the idea then was, well, maybe ARC is being released from a cell that makes, that is transcribing ARC. Those are the cells that undergo LTP. Those are the cells in a circuit that basically they get it, they look like they're um, um, being released from those engram cells, the so-called memory cells. And so then we thought, well, maybe this is a negative signal to other cells that may not be active during that encoding event, but actually, are
Oh,
close. And so at least in culture, we can see that when they get released, the ARC vesicles, they can get transferred to neighboring dendrites, and then they actually affect the AMP receptors. on those.
And did you co-culture neurons from wild type and knockout mice, ARC knockout mice, and then how did you label the ARC and see that it's transferred in these vesicles from one cell to another?
Yeah, so we used knockout neurons, and then we sparsely transfected back in ARC into them. And so then we were able to label the cells that had transfected ARC, and then the rest of
Ah,
the
I
cells
see.
were not.
Yeah.
We want to do wild type versus knockout transfer, but it's difficult because we haven't got a way. It's just difficult to label the two different populations. So this is the way we sort of did it. But
And have you, so these extracellular vesicles I actually did a podcast on that with Dimitros Kapagiannis, who I told you about his work with isolating EVs that maybe are coming from neurons from blood. But um, so have you interrogated these vesicles that contain ARC for their molecular composition, for example, proteins, RNA,
yeah,
in relation to vesicles that don't have ARC or something like that? Is there any
yeah.
specificity thing unusual about those vesicles just compared to any other?
Yeah, no, that's a good question. I mean, that's actually been something that's a little harder, been harder to sort of get at than we thought. So
Yeah.
we know that an arc is released. It's actually a subset of vesicles at any given time point. So, and we haven't had a good way of just purifying the arc only vesicles to get at the cargo. So we have tried what you suggested, which is to compare arc knockout vesicles to wild type. And we had some differences, but it's just noisy. And so I would say the only cargo that we're sure of is ARC's mRNA. So the ARC transfer of its own mRNA seems to be important. And then we can talk about this disease angle where we think that actually ARC is binding tau and bringing tau into these vesicles.
Okay, and you haven't mentioned this until now that ARC binds to its own, the ARC protein binds to its own messenger RNA.
Yeah.
And does it also bind to others?
So in it's another sort of, you know, question that I thought we'd answered. So we know that arc mRNA is the main cargo, but there could be some non-coding RNAs. We're just not sure of what those are. So yeah, we're still struggling to get there.
Okay, so this is kind of cool. So these vesicles with ARC, and they have the mRNA bound, and they're transferred from, say, the dendrites of one neuron when that synapse is activated to whatever adjacent dendrites could be of the same neuron, actually, I guess, or
Right,
other neurons, or even presynaptic cells,
right.
and then that ARC mess can the ARC messenger RNA, have you shown that it gets into another cell
Yep.
and is translated into ARC protein in that other cell?
Yeah, we shown that, and in fact, the this when we see this transfer into neighboring dendrites and the effect on the AMP receptors, if we block translation of ARC in those recipient cells, we don't see the effect.
Wow.
Actually, I mean, it's kind of surprising, and the whole thing is weird. I mean, I had a bet with my student because I was like, well, maybe the capsid, which we know has like 100 or so monomers or whatever, is a good way of just getting a bunch of ARC into the cell. Why would you need then translation and et cetera? But there's something special we think about that local translation.
Yeah, and okay, so then, so this ARC protein is like these coat proteins on a lot of viruses,
Right,
right? That form the structure around the content, the RNA or DNA, I guess, could be either one depending on the virus.
right.
Um, and right, and how far, well, this is getting really complex, but you can imagine like the mRNA from one cell gets into another, then it's translated, binds with more messenger RNA, then it's translated. So you could have like a chain
Yeah.
reaction effect.
So that's why we think there's something special about the locally translated population of ARC, where that is the population that's important for the AMP receptor trafficking, the synaptic function. And there we think there's either PTMs or again sort of different binding partners that prevent it from making a capsid, but recruit the AMP receptors. So there's these two populations. So we don't think it's, like it gets transferred and then makes more capsids and then it gets transferred again. We think that this is sort of a, um, uh, like it's not like a replication event.
Now,
Um,
one thing is the membrane of cells, like things are not uniform, right? Like there's certain regions where receptors are, congregated, I guess you'd say, like glutamate receptors, or it could be growth factor receptors. And those regions of the membrane have different lipid composition often than other regions. There's something called lipid rafts where, uh,
right.
and do you know if the arc, like it tends to go to like lipid rafts, high cholesterol regions of the membrane or something like that?
Yeah, so we haven't got to that point. So what we have observed with some live imaging experiments is that release happens in the dendrites, and actually it may happen from these filipodial-like projections.
Huh.
Um,
Hmm.
and so this is through this other protein called IRS-P53, which is a bar domain containing protein that binds to other membranes, and as it also is involved in making filipodia and spines. So right now we're trying to nail exactly where the release happens, but we suspect it could be this unique sort of filipodial like projection, but we don't. If that's the case, then it would have, as you sort of say, unique membrane and transmembrane proteins. So yeah, lots of
So
symbiosis.
so my postdoc work, I was studying growth cones, right?
Oh yeah,
And the regulation by glutamate. And me and Holly Klein pretty much at the same time were finding that glutamate uh, affects motility of dendrite growth cones and it's important for synaptogenesis. And so the actin filaments, right, as Filipodia are, you get polymerization of actin filaments.
exactly.
So
Yep.
there's some linkage between the arc in either monomeric or multimeric form and the actin cytoskeleton.
We suspect that is the case, and actually, OX stands for activity cytoskeletal protein, but
Yeah, that's right.
uh, the IRSP53 protein also has downstream effector actin domains, and if you block those domains or you mutate those domains, then you don't get release of OX. So we do think probably actin is involved in that process.
Okay, so let's get to, we're about halfway done. So that's what, well, that's 20 years of your life about,
Yeah,
so far,
yeah, yeah, yeah.
in a half hour. Okay, neurodegenerative disorders. You've
Right.
done some really interesting work there. And so go ahead and start talking about that. And that's something you're doing a lot on now, I gather. I
Yeah.
mean, there's this, uh, for people who don't know, beginning, I don't know how long ago, not that many years ago, there started these what are called preprint servers, where people can put up their findings that are available to anybody to see before they've been peer reviewed, right?
Right.
So so this is up there, right? I was looking at it.
Yeah.
The Tau story. Talk about this.
Yeah, so I managed to get this nice grant from the Chan Zuckerberg Initiative to sort of
Uh,
look at neurodegeneration. And I had no, there was this idea that the spread of pathology is a common denominator for all neurodegeneration neurodegenerative disorders, where toxic proteins misfold and then they accumulate in a cell, but then they could get released or secreted either through some sort of secretory pathway or some of these autophagy or lysosome pathways, and that the secretion of these toxic proteins then allows or propagates the proteins to other cells. And there had been some evidence that extracellular vesicles were part of this. Um, so I was like, well, just as a hunch, maybe ARC is somehow involved in this process. I had started a project in Paul's lab looking at amyloid and A beta, and we had crossed the ARC knockouts to an amyloid model, a presenilin APP mouse. And so we had a paper on that where we had shown that when you cross the ARC knockouts, you actually get less plaques, and that this could be through an activity dependent A beta pathway. But I was sort of interested in tau, partly just to see if there was something there. And tau propagation, there was clear evidence for tau in extracellular vesicles. And so we took knockout neurons and did this tau transfer assay that Brad Hyman's group had developed. And lo and behold, when we did this transfer assay in the knockout background, there was very little transfer. In fact, I mean, the phenotype was so dramatic that I was like, I had to tell my student she had to repeat it a couple more times to make sure that this was a real result.
So the tau transfer, how did you do that? So you have ARC knockout mice with no ARC in their neurons, and then somehow you put pathogenic human tau, I guess,
Yeah,
in some neurons.
exactly. So we used like an AV approach that's a sparse AV approach to transduce neurons that would express human tau, human mutated tau, and GFP, but there was a linker, a 2A linker. And so the idea was that the neuron would produce equal amounts of GFP and tau, but they weren't fused, they were split. And so you would have both GFP and tau in the transduced cells. And then if tau was transferred to other cells, there would not be GFP, it would just be tau. So it gave you a nice way to sort of look at this transfer. And this was, again, Brad Hyman's group's development. So we did
And
that first in culture.
which neurons using the VIA AV did you express that
Yeah,
time?
so we did this first in hippocampal, primary cultured hippocampal neurons.
Okay.
And then Matali did this in vivo, where she injected the same virus into the entorhinal cortex. Um,
Uh, so when I got my PhD at University of Iowa, and Brad Hyman was there.
oh wow,
Yeah, he trained with a guy named Gary Van Hosen, and Brad had this kind of, well, famous, if you will, in the Alzheimer's field paper where he provided pretty strong evidence by doing post-mortem analysis of people who died with different stages of dementia that you see the tau pathology first in the entorhinal cortex.
right,
So that's why they did it in the entorhinal cortex.
right, exactly.
Yeah.
Um, so yeah, so we did the same thing in vivo and saw the same dramatic effect in ARC knockout mice that the transfer in vivo was also way less than wild types. So that
Okay, so then now I'm joking here, but so to treat Alzheimer's disease, we just need to knock out ARC, and the people will be able to learn things, but they won't be able to remember things, and they won't have any tau pathology.
right. So so you obviously don't want to do that. I mean, it would be
No,
so
I know.
yeah, so it's tricky. I mean, the hope here is that so anyway, so I'll continue the story and then we can talk about sort of maybe translational
Yeah.
aspects. But so that phenotype was clear, and so we wanted to figure out, well, why? And the obvious hypothesis was that ARC was somehow important for packaging tau into vesicles. Then it's the vesicles, the vesicular tau that's efficient in getting transferred. And so that we isolated extracellular vesicles from brains, and we took these tau transgenic mice that overexpress human tau, mutated human tau, that develop tau pathology pretty young. And so we looked for tau in vesicles from the brains that were either wild type background or crossed to the ARC knockout background. Um, and yeah, and what we see is that indeed there's a fair amount of tau in vesicles that we extracted from wild type brain transgenics, but then in the ARC knockout background, there's almost no tau in the vesicle population. There is still naked tau, so there's two populations of tau that get secreted. There's membrane bound tau, vesicular tau, and there's the naked tau. And so ARC seems to preferentially package tau into vesicles. So without ARC there, you don't have the
Huh.
vesicular population.
And is there any direct interaction between ARC and tau, a physical interaction?
Yeah, so then we looked at that, and there is. And actually, it's kind of interesting. We're still trying to figure out how exactly that interaction works. But we could co-IP it. We can GST pull down the purified proteins. We're pretty sure that there's a direct interaction. We even in the preprint have a collaborator that's try to model the interaction with biophysical models, and it's interesting. And we're still trying to figure out what that interaction actually means. But the outcome of that was that we think that ARC directly binds tau, but the way that they bind still lets ARC form a capsid. And of course, we don't think tau protein is being put inside the capsid, but it's being brought into the vesicle.
Yeah. Okay. And so tau, this microtubule associated protein, under healthy conditions, it's mostly in the axons, right?
Right,
And ARC is mostly in the dendrites. But then
right,
it's thought in Alzheimer's disease, there's tau becomes hyperphosphorylated. It comes off the microtubules, maybe self-aggregates, and it definitely is present probably towards later stages of the degenerative process in dendrites. But
right.
how are you visioning this in terms of the early versus late events in the tau pathology and the degenerative process?
Yeah, so we're still trying to figure that out, but our initial sort of hypothesis here that sort of there's evidence for this already is that early in the disease, or early as the sort of cells are getting pathology, there's hyper-excitable circuits. So there's
Yeah.
more activity, and there's sort of these subthreshold seizures. Exactly. And so that initial event, and in fact, other people like Leonard Mookie has shown that ARC induction can be massive when you have these subthreshold events. So it's
Ah,
possible that early on, there's synaptic dysfunction, there is circuit dysfunction, but it's hyperexcitable, and that would accelerate this process. But then late in the disease, as the cells are dying or are dead, then you would expect there's no activity or low activity, and so you wouldn't see. So we do think that the contribution here happens early. Um, yeah.
and is Arkin GABAergic? Because it's been mostly studied in the glutamatergic excitatory neurons. Is it also
Yeah.
in GABAergic, and does it
So
have a
so
role?
in cortex, it's not transcribed, we think, in an interneurons or inhibitory cells. But we don't know if the transfer events
Ah,
could happen
yeah.
to inhibitory cells. And the big challenge now is that most of our work initially has been done in culture, and so now we're trying to build tools to identify in vivo, what are the donor cells, what are the recipient cells, and what exactly is the spatial, specificity there.
Uh, yeah, I'm thinking. So there are certain uh, so extracellular vesicles can come in different flavors, right? There's kind of two major classes that I'm aware of. and these are small, like 100 nanometers in diameter, so they're quite small. They can come
Right.
either from these what are called multivesicular bodies, that are essentially um, a, a big membranous sphere with a bunch of small spheres in it, and then that fuses with the plasma membrane of the cell and releases those small exosomes or
Right.
extracellular vesicles. But then also you can get the plasma membrane can kind of just form blebs, or
Right.
do you know which of those two or both ARC is mainly released in?
Yeah, so we think under activity conditions like LTP, ARC is being released directly from the cell surface and it doesn't go through this exosome or multivesicular body um, pathway. And this is what retroviruses do as well for the most part. But even
Uh,
the retroviruses, even HIV, in some cases can, depending on the cell type, go through a multivesicular body release mechanism too. So we haven't ruled out that ARC does go through an exosome pathway, but our live imaging experiments and this IRSP53 mechanism we think happens through this direct budding. And
okay.
so it's a unique sort of um, extracellular vesicle as opposed to the exosomes.
Okay. So then, right. And did you do any? I'm looking at your abstract now from the tau paper. Did you do any behavior?
We didn't do any behavior, partly because, or not yet, partly because, the confound here is that when you knock out ARC, there are also going to be cognitive changes or deficits.
Uh,
Um,
yeah.
so we need an assay, both behavior and plasticity wise, that we know ARC is intact, is intact in the ARC
Yeah.
knockouts. So
Yeah.
yeah, so that's a little bit challenging.
Yeah. Oh, right. But since it since ARC seems to be important for the transcellular transfer of the pathogenic tau, shouldn't the and so the tau, sorry, the ARC knockout mice, they learn well.
Right.
Um, and in that tau model, do those mice have problems in learning and acquisition? And does ARC knockout recover that ability to learn?
Yeah, so we haven't looked at that closely, but you're right. That's where we could go is to try and find these learning deficits in the tau transgenic that are intact, like intact in the just in the ARC knockout. But yeah, so
That's a
that's
good project for even an undergraduate, actually, if you're
yeah,
just
yeah,
doing behavior.
yeah,
It
exactly.
gives them good experience handling mice and
Right.
kind of a neat thing for them.
Yeah.
Okay, so you're doing a lot of collaborations. So I looked through your publications and you've got a lot of collaborations with people who are looking at different aspects of neuroscience mainly. Like, for example, I saw this one where um, the collaborators and with your help find that ARC plays a role in vasodilation.
Oh yeah, yep.
I have this, so I have this, and I didn't know I had this for decades. Um, I have something called primary erythromelalgia,
Hmm.
where um, I have a mutation in a voltage-dependent sodium channel, NAV 1.9, that's highly expressed in nociceptive neurons, pain-conveying neurons, but it's also in those small uh, uh, nerve fibers that innervate the blood vessels. And so I get these episodes of my feet get really red
Right.
and painful. It's kind of a neuropathy condition.
Hmm.
So I was very interested in this from the standpoint of myself. Do you want to talk about that? So essentially, they find in the ARC knockout mice there's some abnormality in the control of
Yeah.
the blood.
Yeah, this was work from Zach Campbell, and I was, barely a collaborator, but
Yeah.
really interesting science. So he was interested in these pain and toxic, pain and sort of noxious stimuli and how they signal from the skin to the dorsal root ganglion, the peripheral neurons. And he had noticed that arc would go up as some of these other immediate early genes as well would go up in the dorsal root ganglion neurons after, having this sort of chronic or acute noxious event in the skin. And so he was interested in, well, if arc goes up, what's it doing? And so they took the arc knockouts and did this experiment where they would inject a toxin in the paw that would induce inflammation and vasodilation. And what they saw was sort of interesting in the knockouts. There was increased vasodilation. There was sort of decoupling from the signal to this from the skin to the vessels. But the most interesting thing was that he could restore this signal by just injecting arc vesicles back into the knockout. Um, so that's as far as we got, and then he actually moved recently to a different university. So we haven't picked it up again, but um, it suggested that the hypothesis was that the peripheral neurons, even these dorsal root ganglion neurons, were releasing arc, and they were carrying a signal to the skin. But what that signal was, what the cells are in the skin that are actually taking up arc, we don't know.
Okay, Alzheimer's disease, pain syndromes. There's also, I guess, early evidence actually related to, I guess, autism spectrum disorders.
Right.
Mark, you want to briefly talk about that?
Yeah, I mean, so um, ARC is kind of interesting because it's an immediate early gene, but it's not a transcription factor, unlike a lot of these, like FOS. And it turned out that in a lot of these intellectual disability disorders, these monogenic disorders like Fragile X syndrome and Angelman syndrome, um, the pathways converge on these activity-dependent processes, including ARC. And so we think that ARC is just one of many um, activity-dependent genes that are dysregulated in those disorders. So, for example, Fragile X syndrome. Fragile X is an RNA binding protein that then regulates translation, perhaps even local translation. And without Fragile X protein there, you get this increase in protein synthesis. And ARC is one of those proteins that's just regulated, goes up. And then Angelman syndrome, there was evidence from Mike Greenberg's lab that UB3A, which is the protein that's mutated, it's a ubiquitin ligase and it actually degrades proteins. And so similar thing there. So ARC levels go up in UB3A knockout neurons, potentially because they're not degraded. The
Yeah.
protein's not degraded. So the outcome is basically, and this is kind of similar to many synaptic proteins as well, is that if you have too little or too much of the protein, it's bad.
Causes trouble. Yeah. Okay, very interesting. Any other collaborations that you're,
Um,
I mean, you're doing a lot, but
Yeah,
what,
so we got interested in, the evolution side of things a little bit of these genes. And I've also been interested in all the other genes that have similar biology, and so we've been branching out a little bit there. And in fact, one of them is called PNMA2, these PNMA proteins, which have not been well characterized, but this PNMA stands for paraneoplastic antigen because there's autoantibodies generated in rare cancers where it's thought that these autoantibodies are a sign of an autoimmune disorder where there's actually neurological symptoms. And but it wasn't clear why this family, this PNMA protein, is actually involved in an autoimmune um, disorder. And it turns out, well, I mean, of course, then we thought, well, maybe it's because it makes these viral-like capsids, and indeed, this PNMA2 protein makes these beautiful capsids that seem to be really quite immunogenic.
huh,
So we published that paper earlier this year that sort of suggests that if some of these capsid-forming proteins that are normally expressed in the brain get expressed really high levels outside the nervous system, they can induce this immune response. Um, But so yeah, we're sort of also looking at the normal function of this PNMA2 protein. Um, and then another collaboration that we've got right now is actually looking at the fly genes, the fly arc genes. But it turns out the fly arc genes are not direct homology, they're not ancestral to the mammalian arc. They're actually independently evolved, repurposed from different retrotransposons. But the wild thing is that the fly protein also expresses at synapses and it's also released in vesicles. And that was shown by Vivian Budnick's group at the same time as we were looking at the ARC version, the mammalian version. So we wanted to know whether the fly genes, the ARC genes, were involved in learning and memory. So we're collaborating with a group here at Utah, Sophie Caron, who's a Drosophila biologist, and knocked out the ARC genes, DARC1 and 2. And indeed, we see this learning defect or learning phenotype. Um, Actually, they learn better. Well, they learn reward kind of learning better and punishment kind of learning worse. And we think this is because of the circuit that's regulating in the fly brain. Um, but the remarkable thing for me is that these are very different evolutionary histories,
Yeah.
but somehow evolution has decided that this viral-like biology, the capsids, are important or solve some problem in the nervous system that's better than others. But what that is, we're still trying to figure out.
And are there arc-like uh, proteins in organisms that don't have nervous systems? So, I mean, going way back uh,
Yeah.
in evolution.
That's a good question. We know that there are these, the retrotransposons themselves are ancient. You see them even in yeast, and actually Josh Dubnow has shown that the active retrotransposons themselves can be, important for sort of some normal function in the fly. Um, but whether there is a similar, repurposed gene in non-nervous system containing organisms, I don't know.
There had to be at some point, there's a big adaptive value of retaining these arc-like proteins,
Yeah,
and somehow, I mean, it's a speculation, but they had to, over time, right, that there had to be a process, like a stepwise process whereby they took on a role in synaptic plasticity.
right.
Um,
Yeah, and
yeah.
so the interesting thing there, so what we don't know for the fly genes is what are they actually doing in the cell? Do they also have a synaptic function? And we don't know. They lack the N-terminal part of the mammalian protein where we think some of these interactions are happening with
uh,
the synaptic
something.
protein. So it could be that they're doing different things, but that the transfer of their RNA is the commonality there. Um, but yeah, fascinating stuff. Like we there's lots of questions that we have to figure out.
And what about translational research? Is there any way that you can kind of explore or take advantage of what you found with ARC and its role in, say, tau pathology or so on, or you know,
Yeah,
its role in oh, so as you know, my lab did a lot of work on intermittent fasting, exercise effects, so there's certain environmental enriched environments, right? So ARC's upregulated with probably, well, definitely with enriched environments,
yep,
exercise.
yep.
I don't think anyone's looked at intermittent fasting.
Not directly. I have a colleague here that has looked at just has done like an omics approach to this, and he said he does actually see ARC being upregulated during fasting or after fasting.
Kind of
Uh,
makes sense from going back to evolution, right? That when you're in a food deprived state, you're actually, I saw one paper on like a model of foraging, right?
right, exactly. Yeah, Chris
Right.
Craig. So that was the that's my colleague here who we have been working with, and yeah, he thinks that ARC does get induced by caloric restriction.
Right. So I would hypothesize and art took on some role that's fundamental for survival, maybe in acquiring food, being able to, you got to have good learning and memory.
Yeah, exactly.
And
Yep.
also exercise. I think today, do you have a hard stop at in 4 minutes or can you go a little over?
Um, yeah, kind of, but we can go a little bit longer.
Okay. So one thing I'm worried about, like kids today not getting out in the woods and stuff and moving through their environment while they're learning things. We kind of just sit in one place and
Right.
learn things. But I think we learn things, or we do a lot of at least creative and intellectual thinking when we're moving. At least I do.
Yeah,
Like running, I used to run a lot, or walk, even walking. A
right.
lot of people find I'll be writing a paper, and I've worked on it for a couple hours, and so I just go out and walk for a half hour or something, and kind of think about what I've written and
Right.
what. So, yeah, art's probably playing some really important roles in these kind of evolutionarily important things
Yeah.
that are happening in the brain with exercise or
Yeah,
fasting.
exactly.
Yeah.
We haven't looked at directly in mammals, but yeah, I think you're right. And actually, that fly phenotype with the sugar reward, we suspect it's something to do with signaling specifically for sugar,
Ah,
which would make sense in that sort of evolutionary sort of idea.
okay, so now you got to look at the gustatory system and
Yeah,
olfactory system and
yeah, exactly. At least in
okay.
that one.
All right. Anything else you want to say?
No, I guess, oh, the translational question. So
Oh,
one thing we did have done, I did, is this idea that perhaps we can actually use these endogenous capsids for gene delivery. As you know, like gene therapy, the main way to do that is to use modified viruses, which have their own issues. So I co-founded a company to sort of look at this called V&V, which stood for virus, not virus. And more recently, it got taken over by a company that Fong Zhang had started called AERA. So that's a direct translational outcome of sort of this biology is to perhaps use these endogenous capsids that you could modify a little bit for cargo.
so for that, Jason, so you synthesize ARC and uh then you mix it with RNA.
RNA, yeah,
Yeah.
different RNAs. And then the idea would be that perhaps, there's unique aspects of the capsid that are important for allowing it to get into neurons versus other cells, for example. And as you know, it's been difficult to get to sort of have neuronal specificity for uptake of these things. So that's the hope, or the idea.
And then how would you do brain delivery then?
Yeah,
How
so
would
that's all gonna that's also stuff we have to figure out
yeah.
still that they are doing in vivo, which is, like just can you get blood-brain barrier crossing and stuff like that.
Right, but you said that the your colleague who did the pain stuff found that you could so in that model you could just add that could be maybe a initial screen
Yeah,
for doing that.
yeah.
Ah, I'll volunteer for. Okay, so here's what we're going to do. You get what do we want? We, we want actually we want to do RNA interference. So I want to knock out my NAV 1.9 gene
Right.
in my
And peripheral neurons,
in
yep.
my peripheral neurons. So my foot's here, right? So I volunteer you can inject in my foot and
I'll let them know.
and we have one foot is the control for the other. So we'll just
Right.
do one and then all right.
Sounds good.
Actually, I talk with people. There's people at Hopkins trying to develop drugs. They're expressing my mutant channel
Oh yeah.
in cells and screening. But I also have there's a professor at Hopkins who did a postdoc with me, and she could do a AAV with RNAi
Right,
or something like that.
right.
But I can't get anybody to inject into my dorsal root ganglia.
Yeah, I
So,
assume that there's some IRB issues there.
yeah. Okay, but anyway, interesting. There is translational potential. I'll be interested to see how that
Right.
develops.
Yep, exactly.
Okay, well, you're in a nice location there. You must have learned to like the outdoors in New Zealand, and then
Yeah, exactly. No, we love living up here in the mountains, and I ski and do a lot of hiking and backpacking, so it's great.
yeah, it's a great place. Okay, Jason, it's good to see you.
Yeah, thanks, Mark. It was great to see you.
Yep, take care.
Cheers.
Bye.
Bye.